Exploring cells. An introduction to observing cells with a light microscope. Today we will describe why cells are called the building blocks of life. Name the parts of a light microscope. And we'll explain how to use a microscope. So please watch this video till end. And subscribe my YouTube channel Khalil's Quiz Zone for more videos. A cell is the smallest unit of a living thing. It is the basic building block of all living organisms. You can see the shape of animal cell and plant cell here. A living thing may be made of one cell, a unicellular organism, or many cells, a multicellular organism. Humans have about 37.2 trillion cells. And humans are multicellular organisms. Euglena is a unicellular organism. Robert Hooke is considered to be the father of cytology. Robert Hooke studied cork cells under a simple microscope and discovered cells. Cytology deals with the study of the function and structure of plant and animal cells and their characteristics. Robert Hooke coined the term cell in 1665. His biggest scientific achievements was the invention of the modern microscope. Hooke is also credited for the invention of the compound microscope. Cell sizes can vary from about 0.001 mm to 0.1 mm long. To see cells up close, we use a light microscope. It provides a magnified image of small structures and samples. To use the microscope well, you must be familiar with its main parts. Eyepiece, coarse adjustment wheel, fine adjustment wheel, rotating nose, objective lenses, stage, aperture and mirror, light source are the main parts of a light microscope. If you have never used a light microscope before, this handy guide will explain how it's done. Step 1. Turn the light microscope on, move the mirror to reflect the light up towards the eyepiece and check that the aperture is wide open. Step 2. Turn the revolving nosepiece so the lowest objective lens is pointing directly down. Step 3. Place your specimen onto the stage. Step 4. Look into the eyepiece and slowly rotate the coarse adjustment wheel to bring your specimen into focus. Step 5. Slowly rotate the fine adjustment wheel until you obtain a clearer image of your specimen. Step 6. Examine and explore your specimen, holding still or moving the slide slowly with your thumbs. Step 7. Once you have seen enough, switch to the medium and the high-power objective lens. Use the fine adjustment wheel to bring the specimen into focus as necessary. Light rays go through the aperture, to the specimen, to the objective lens, then to the eyepiece and into the eye. But remember, light rays bend as they pass through the lenses, allowing the image to be magnified. The main idea of a light microscope is for the light rays to go through the specimen. So, specimens must be thin enough for the light to pass through.